This is Mr. Sachin Kharade, Assistant Professor, Kekewa College of Agricultural Biotechnology, Nasik, going to deliver a topic on cytoskeleton. So this is the basic structure of cell, which having the different cell organelles like cell wall, plasma membrane, then endoplasmic reticulum, nucleus, ribosomes, Golgi apparatus, then the mitochondria. So, from among these cell organelle, one of the most important part in the cell, that is cytoskeleton, which is comprises microtubules, then the intermediate filaments and the microfilaments. So we will turn towards the basic introduction of cytoskeleton. First of all, we should know what is cytoskeleton. So it is a complex network of filaments and microtubules which form a structural framework known as cytoskeleton. So the skeleton and muscle of the cell. So it is behave like a skeleton and muscles of cell. It is also responsible for segregation of chromosomes into two daughter cells at the time of division. So these figure or photographs which are interpreted here, that is cytoskeleton. So the plasma membrane and these cell organelles which having the interconnection between each other by this cytoskeleton. So it is the microscopic picture of cytoskeleton. So the cytoskeleton is built on a framework of three types of protein filaments. First one is the intermediate filaments, second one is the microtubules, and third one is the actin filaments. So this microtubule, which is 25 nanometer in diameter and which is tube-like structure. So next one is the actin filaments, which is 7 nanometer in diameter, and third one is the intermediate filament, which is 10 nanometer in diameter. So intermediate filaments, that is, they have great tensile strength, which is diameter in about 10 nanometer and made up of fibrous intermediate proteins. So it is composed of two antiparallel diameter. First one is the we can say, if it is one, that is it is monomer. If it is two in parallel dimer, we can call it as a dimer, and if three, that is tetramer. So this is the basic structure of monomer, dimer and tetramer. So types of intermediate filaments, that is keratin filament, second one is the vimentin filament, third one is the neurofilament, fourth one is the nuclear lamins. So disorders of intermediate filaments are as follows. So epidermal lysis bullosa simplex, we can call it as a EBS. Second one is the amyotrophic lateral sclerosis, that is ALS, and third one is the progeria. So these microtubules is also one of the important part of the cytoskeleton, which is long, hollow, cylindrical, made up of tubulin, which is 25 nanometer in diameter. It will provide internal shape and support to the cell, also involved in nucleic and cell division. It having the two types of tubulins, which are found in alpha tubulin and beta tubulin. So this is the side view of tubulins, which having the 17 nanometer or in the 25 nanometer. This is a cross section of tubulins, and it is protofilament, and which is this one is the diameter. So the structure of microtubules which are given here. So first one is the microtubule structure, which is second one is the microtubules in an axon. Uh, First one, this is C1 is the different types of microtubules. First one is the singlet, then doublet and triplet, which having the negative and positive end. So cilia and flagella, it is also important part of cytoskeleton. So cilia and flagella are hair-like structure made up of microtubules and converted by extension of plasma membrane. So cilia and flagella moves liquid past the surface of the cell. They create current of obtaining food from the aquatic medium. On epithelial cell lining of respiratory tract, huge number of cilia are found. So this is the basic structure of cilia, which is hair-like structure, and flagella. This is the structure of flagella, which having the 1 micrometer. So 9+2 arrangement in the structure of flagella. And so drugs that affect microtubules, that is Taxol, then the colchicine and vinblastine. So these are the drugs that affects the microtubules. So actin filaments, which is present throughout the cytoplasm of eukaryotic cell, present just beneath the plasma membrane, therefore supports the outer layer of cell. So it is responsible for movement, especially involving in the cell surface. Can form structure like a microvilli on the brush bound bordered cells lining the intestine. So actin filaments grow by addition of actin monomer, but in order to stop more of addition of actin monomer, proteins such as thymosin and profilin bind to the monomers. So it is the dynamic structure of these actin filaments. So movement by actin filaments are as follows. First one is the cell phases out protrusions at the leading edges. Then these protrusions adhere to the surface. And third one is the rest of the cell drags itself forward on this anchorage point. So first stage, that is protrusion of the leading edges, then the adhesion, then the movement of the cell body. So this is muscle contraction. Myosin along with the actin was first discovered with the skeletal muscle. So myosin 1 having only one head and a tail, but myosin second having two ATPase head and a tail. So muscle myosin belongs to the myosin second. So this is the basic structure of cytoskeleton of the muscle. So this muscle, then the fascials, then muscle fibers, then the myofibrils, and the thick and thin filament, which are shown in the figure. So each muscle fiber is a single long cylindrical muscle cell. Second, scrollal cell membrane. So sarcoplasm, cytoplasm with many mitochondria and nuclei. It has microfibrils. Microfibrils are separated into the components called as sarcomers that contains thick filament and thin filaments. So thick filaments of microfibrils are made up of protein, that is myosin. Thin filament of microfibrils are made up of protein, that is actin. and the organization of these filaments produces striations. So this is the basic structure of thin filament, and this is the basic structure of thick filament. Thin filament, which is made up of actin molecules, and thick filaments, which are made up of myosin. So sarcomeres extended from the Z line to Z line. So one first bands, that is light band, made up of actin and anchored to the Z line. So A bands, that is dark band, made up of overlapping thick and thin filaments, and in center of A band is an H zone consisting of myosin filaments only. So, sliding filaments theory of muscle contraction, the myosin cross bridge attached to the binding site on the actin filaments and bends, pulling on the actin filaments. It then released and attaches to the next binding site on the actin, pulling again. So this is the procedure. So with this small introduction and structure of cytoskeleton, so thank you for listening.